I'm just going to talk to you a little bit about some other workflows that you might imagine and how we think the MISeq i100 could actually make an impact in this specific field. My name is Jennifer Mummery and I'm a field application scientist here at Illumina. And one of the focuses that I have in the clinical team is infectious disease and microbiology, and that's why Jen asked me to come today and speak to you about that. So we know that especially in the field of microbiology and infectious disease, time to results and accuracy are really critical. And as you've heard already from some of the other speakers, these are really important advances that we've made with the new MISeq platform. So I hope I can show you some ways why we think this will be especially relevant to that type of application. So we know that NGS, we just heard how NGS can be applied to various different types of analysis in microbiology and infectious disease. One example will be for managing outbreaks, when and where they happen. We know that causes of outbreaks are various. We can have zoonotic causes, for example, Mpox or COVID or avian flu. Climate changes are bringing us more and more infectious diseases, for example, mosquito-borne diseases like dengue or Zika. We also have foodborne pathogens that impact patients like Listeria, Campylobacter, or E. coli. Whole genome sequencing is actually a key tool we can use to understand these pathogens at the genomic level and really take that information as part of a package for informing public health decisions. We also know that wastewater genomic surveillance is becoming increasingly more important, be that wastewater from whole cities, from hospitals, from aircrafts, from dorm rooms. We can imagine lots of different applications where we can use wastewater surveillance also as an early warning sign for endemic surveillance and also for new emerging pathogens. But we can do more. So NGS can also be used to fight high-risk bacterial pathogens and to help control healthcare-associated infections. We know that bacterial priority pathogens we have a high focus on that from many different hospital settings, but even the World Health Organization has maintained and this year made an update to its priority pathogens list, telling us that this is really something that the world needs to start paying more attention to. Whole genome sequencing can be useful for studying the genomic epidemiology at the individual hospital level. It can be helpful to understand local resistance epidemiology and the strains that are circulating and present in that local environment, but it can also help hospitals to define their antibiotic stewardship. So in which cases, under which circumstances, for which patients with which infections would antibiotics be prescribed. Healthcare-acquired infections, or HAI, is a problem that we are paying more and more attention to. And in order to control those types of infections, we need fast and accurate genomic information. Whole genome sequencing can be used to compare the strains and identify the source of the contamination as well. Antimicrobial resistance, we heard also Tim talked about antimicrobial resistance and virulence markers as well. We know that's also a major public health priority. There are estimates about the global burden of antimicrobial resistance, which estimate that by 2050, almost 2 million people, most of them, or majority of them in this age 70 and over, could actually die from drug-resistant infections each year. The United Nations also maintains a list of and a working group on antimicrobial resistance, and they've just had a high-level meeting on AMR in September of this year. We can really use NGS to tackle antimicrobial resistance by surveying the markers that are circulating and monitoring as well. And we have many tools that can help customers in doing that, including our capture-based enrichment panels like the UPIP, which contains more than 3,700 antibiotic resistance markers. NGS can also push further and help you also to understand unknown samples or unknown things, emerging pathogens, by using a more broad-based application like metagenomic or metatranscriptomic profiling. In clinical metagenomics, for clinical research, various different infected sample types could be used as an input. be that respiratory, prosthetic joint samples, or meningitis samples. And it can also help us find answers about non-cultivatable pathogens or other unusual pathogens that might not be included in routine molecular panels. Microbiome analysis is also a hot topic in this area, and shotgun metagenomics opens a lot of new applications. We can imagine analyzing and studying the effect of changes in oral dental microbiome, in the vaginal microbiome during pregnancy, or in the gut microbiome. Metatranscriptomics pushes it even that much further and lets you study also the host response to infections, and opens the door to even things maybe we haven't started to consider yet. So the MISeq I100 series can, as Jen showed you the slide already, the beauty of the platform is that it allows you to do very targeted things that you could already do on the on-market MISeq system, such as 16S amplicon sequencing or small whole genome sequencing. But with the same instrument, you'd also be able to do broader panels or more high-depth sequencing requirements, such as targeting gene sequencing across single or multiple different organisms, moving into the metagenomic space, and even into the transcriptome profiling. So I'll just present with you a few of the illumina solutions for microbiology and infectious disease. And we can keep in mind that within infectious disease and microbiology, we can go from very focused applications, for example, whole genome sequencing of a single microbial isolate, across a broad range of targeted applications, be that with amplicons or with capture-based enrichment panels, or to really wide breadth samples looking at whole transcriptome or metagenome profiling within a sample. So some examples of these kits that we have as ready-to-use on the market kits. In amplicon solutions, we offer kits for monitoring COVID, so SARS-CoV-2 virus with the COVID-seq kit. Using that same proven chemistry, we launched in the last year the IMAP flu kit, which targets influenza A and B virus family members. And you can also use the same chemistry, but bring your own content in the Illumina Microbial Amplicon Prep or IMAP kit. Here, customers can bring their own content or use published content to study whole genome sequencing of various different pathogens, especially some of the viruses that I've outlined here on the slide. In terms of enrichment kits, we actually have quite a wide range now, targeting different types of pathogens for either identification or for surveillance. For example, for respiratory pathogens, we have the RPIP kit or the RVOP kit. The RPIP kit, which includes identification especially, and RVOP looking more at the whole genome sequencing of respiratory pathogens. Our viral surveillance panel, also released in the last year, VSPV2, has over 200 viruses covered at whole genome regions in order to help you with your surveillance of all sorts of human relevant pathogens. Genitourinary pathogen identification and surveillance is covered by our UPIP panel, and this is also the panel that has our largest range of antimicrobial resistance markers to help you also to do surveillance of AMR. In addition, we have the possibility to use our enrichment kits together with a custom panel, which you can make using a component of existing panels or bringing your own customer-defined panels. Could cover single or multiple pathogens or different antimicrobial resistance markers. It's really up to you. We'd be very happy to help you with our concierge services to look at those kind of custom panels. In the metagenome space, our kits for metagenomics, metatranscriptomics, or microbial profiling, especially with the Illumina DNA Prep kit, is a very versatile kit for all sorts of shotgun applications as well. And each one of these kits has a corresponding analysis app, meaning you don't need to employ bioinformaticians. If you're just getting started and you'd like to have a solution that matches the library prep kit, we're able to provide that to you as well. So speaking about the data analysis, as you've heard, the MISeq i100 series will have a Dragon analysis on board. So both the hardware FPGA chip enabling really fast and accurate results directly on board with the instrument, meaning that it's seamless integration. As soon as the sequencing is finished, the analysis kicks off, and you can get really from pipetting your sample into your kit all the way to the report. On board, the MISeq i100 series will have 4 apps to start with, and for the microbiology and infectious disease space, the most interesting might be the whole genome sequencing app for small whole genomes, but also the Microbial Enrichment Plus, which will support all of our on-market hybrid capture-based enrichment kits. In addition to the the onboard pipelines, the local analysis, you can use our wide suite of applications on the BaseSpace cloud computing environment. So we have their apps that can help you analyze 16S metagenomic samples. We also have one specifically targeted to amplicon sequencing, and as well as additional pipelines. The analysis, I mentioned that it's very accurate with Dragon, but it's also very fast. And here I just show you some comparison in terms of the analysis time, comparing our on-market MISeq using local run manager software compared to the Dragon onboard apps for MISeq i100 series. If we have a look at the small genome, whole genome sequencing app, with the resequencing workflow on MISeq, it takes about 40 minutes for a 2x150 base pair run for one sample, and only 19 minutes for 24 samples with the same 2x150 configuration. So less than half of the time to do a comparable analysis. And our Microbial Enrichment Plus app on board the MISeq i100 is only taking a little bit more than a half hour to do multiple samples of analysis, which would have had to be done on the cloud or using a local pipeline on the on-market MISeq. How does that compute into a total turnaround time from sample to answer? I've given you a couple of examples of potential workflows that you could use. So here I'm showing you an onboard workflow and how that could compare between MISeq i100 with the Dragon onboard analysis or the MISeq on-market platform now. If we were to use the Dragon Microbial Enrichment Plus app to analyze samples sequenced with MISeq platforms, and which had been prepared for library prep with the Viral Surveillance Panel V2 kit. We go from sample extraction and QC through about 9 hours of library prep. Here we've dramatically reduced the sequencing time with the new platform, and the Enrichment Plus analysis is very quick. And we're looking at a total turnaround time of around 35 hours on the existing MISeq platform, sample to answer, whereas that would only take less than half the time, around 17 hours on the new MISeq i100 Plus. You could also use the BaseSpace apps, and one of the BaseSpace apps that we have available is short read strain typing, so SRST2 app available on BaseSpace. Here we have really a direct comparison using the same analysis app, so analysis time is constant, but taking a sample isolation and extraction, putting it through illumina DNA prep directly from the colony in about a 3.5 hour library prep time, and then sequencing 2x300 cycle kits on either the existing MISeq platform or the new MISeq i100 Plus, we dramatically reduce the sequencing time, and total turnaround time goes from 65-hour range down to 1 day. So we really think that this new platform, MISeq i100 Plus, will be particularly relevant for infectious disease and microbiology, where that fast turnaround time from sample to answer and the accuracy is really critical in order to make decisions to support your work. The onboard analysis also results in a per sample report. It would be the same from the BaseSpace app, and just to give you an idea of the information that you get out from that, here's an example of an HTML report from the Dragon Microbial Enrichment Plus app. And this is a dynamic report which gives you a list of the microbes identified, coverage metrics for the samples, quantity if you're using internal controls in the protocol. You'll get coverage plots for each genome, including segments per genes for segmented viruses, and each report includes also quality control metrics for the sample. So taken together, this is the type of information available even directly on the screen of the instrument that can help you take a quick and actionable decision that your labs will need. With that, I'll finish my talk.